Thank you for joining us. I'm Raquel Faria, an internal medicine specialist at Centro Hospitalar Universitário de Santo António in Porto. Today, I'm honored to host Professor Richard Furie and Maria Dall'Era from the Lupus Academy Steering Committee. You might have noticed that on the 7th February this year, the recent phase 3 REGENCY trial of Obinutuzumab use on lupus nephritis was published in New England Journal of Medicine with positive results. As you know, Obinutuzumab is a humanized type 2 anti-CD20 monoclonal antibody, and the phase 2 trial Nobility already had positive results, which had given a glimpse of hope that a B-cell depletion drug would be approved to the treatment of systemic lupus. Since the trials with rituximab had failed, and everyone knows that it really works in lupus. So lupus nephritis is one of the most severe SLE manifestation, and although several drugs have been recently approved to treat it, like belimumab and voclosporin, it still faces several unmet needs, and some patients still progress to chronic kidney failure and eventually to a higher mortality rate. This trial marks a significant step forward in improving outcomes for lupus nephritis patients. Join us as we break down what this could mean for the future of lupus care. Richard and Maria, welcome, and thank you so much for joining us today. Oh, Richard, you were involved in this trial from the very first steps, and you are the first author of this results paper. Can you please just describe briefly the study and where did it came from?
Well, before I describe the study, let me just tell you a little bit about Obinutuzumab.
Okay, perfect.
The story behind Obinutuzumab actually started with rituximab. So we're going back at least 20 years to the Explorer and Lunar trials. And recall, Lunar was a study of rituximab on a background of mycophenolate in patients with proliferative nephritis. And it's true that statistical significance was not achieved between rituximab and the placebo arm. However, numerical superiority of some, that was around 11 percentage points, was achieved in the rituximab arm. But more importantly, there were several signals that rituximab was effective. For example, 7 patients in the placebo arm required rescue therapy, but none in the rituximab arm. In addition, there was a statistically significant reduction in proteinuria at 1.5 years, but not at 1 year, which was the primary endpoint. And to add to this, there were several studies in rheumatoid arthritis, in SLE, and a post hoc analysis of the Lunar trial that concluded that these patients with the best clinical response were those who had the most robust B-cell depletion. So an antibody that could provide greater B-cell depletion was therefore needed. Enter Obinutuzumab, which was engineered to more effectively kill B-cells. This was accomplished through defucosylation, resulting in a 100-fold increase in antibody-dependent cytotoxicity. And in addition, it was capable of greater direct cell death. And it was about 10 years ago that I attended an advisory board at Roche in Basel and was asked whether the Lunar and Belong trials should be repeated with this more potent antibody. And my answer was obviously yes. Now, as you mentioned, the phase 2 study called Nobility was successful. We refined the design a bit and we're quite confident going into the REGENCY study. The major changes were extending the endpoint from 1 year to 1.5 years. In addition, the endpoint was modified a bit. We'll talk about this later. Changing the requirement to maintain a creatinine within 15% of baseline to maintaining an eGFR within 15% of baseline.
Thank you. Maria, you were eagerly expecting these results, as all of us. And for you, what was the most important finding of this study?
Yes, I think we were all eagerly awaiting these results. You know, we've known, as Richard just reviewed, for over a decade that B cells are critical in the pathogenesis of lupus via a variety of mechanisms. And yet we had the LUNAR and EXPLORER trials, which did not meet their primary endpoints, but had signals of efficacy, as was just described. And thus, we were very much eagerly awaiting these results with a more potent B-cell depleting agent. And I was very happy to see that this trial did meet its primary endpoint, as well as several of the secondary endpoints, I think that were very important, and that we saw a significant efficacy at week 76, as well as I mentioned, some secondary endpoints that we can get to as well. So these are very exciting data because, again, it shows us that this mechanism of targeting B cell does work in lupus, as we would expect. And I think as we think about this particular molecule, obinutuzumab, we can think about it in the spectrum of B cell depletion therapies that are up and coming in lupus. And we can think about, which B cell subset are we impacting? What particular B cells are being impacted by anti-CD20, and then put that in line with some of the other therapies that are coming down the road. So I think this is a great step forward in giving us, again, potentially another option. We have to wait and has to go through the regulatory procedures, but potentially another option that we can use in our patients that are living with lupus nephritis.
Yes. Richard, the primary endpoint was designed to be complete renal response at 76 weeks. Why did you choose the definition of complete renal response? And some of the arguments for the 76 weeks primary endpoint you already gave, but can you please just explore it a little bit? And what changed since nobility?
Yeah, there's no question that complete renal response definitions vary slightly from study to study. And so a lot was gleaned from the nobility trial. We moved the primary endpoint from 1 year to 1.5 years because the peak effect size occurred at 1.5 years in the nobility trial. And we further tweaked the endpoint, requiring that the eGFR be within 85% of the baseline value, as opposed to creatinine being within 85% of the baseline value.
Okay.
And the reason for this was the former definition was just too strict. It required that the creatinine not go up more than 15% in baseline. So for example, if one starts with a creatinine of 0.6, as did a little over a third of the cohort in nobility, and it rises to 0.7, you might just write that off as noise. But in the study in nobility, it was a non-response.
Oh, okay.
So we had to eliminate urinary sediment that was kind of left over from LUNAR. So those are the reasons for the modifications.
Thank you. Maria, do you have any additional comment on this?
Yeah, so I think I was also happy to see those modifications. They're evidence-based, as Rich reviewed. We showed, and several studies have been published now to show that urine sediment is not predictive of overall kidney outcome over time, at least the way that we do it in a clinical trial setting, where we don't actually look at the sediment ourselves, but we send the urine for RBCs. And I agree that, also what has been shown in studies that when patients already have some baseline CKD, so they already have a decline in their eGFR, there's more variability measurement to measurement in terms of the eGFR or the serum creatinine, which is why we have to give more wiggle room, which I think is another reason, again, why the CRR endpoint was slightly modified to give a more lax kind of eGFR requirement than the very strict requirement of 15% that was used in LUNAR. So very good reasons to do that for this new study design.
Thank you. Richard, one of the things that I stuck with on the protocol was that in this trial, there was more obinutuzumab in infusions than in nobility, and why it had two arms with at 1 year treatment. Some would be treated with 1 versus 2 doses. Can you please explore it a little bit more?
Sure, you, like everybody else, is asking me that question. So recall that in Nobility, patients received 2 doses of Obinutuzumab at baseline, and then another 2 doses at 6 months. And what we learned from Nobility is that those patients who had sustained B-cell depletion at 6 and 12 months had better response rates at 1.5 years. And what we also learned is that B-cell repletion occurred sometime between 6 months and 1.5 years. The reason I can't pinpoint it is that we didn't collect B-cell information at 1 year after.
Okay.
So with an endpoint at 1.5 years in Regency, it made sense to dose until 6 months prior to the endpoint, just to make sure that patients remain B-cell depleted. As far as the last course consisting of just 1 dose for half the cohort or 2 doses for the other half of the cohort, that was a regulatory suggestion just to get a better handle on long-term dosing.
Okay. Maria, as a clinician, do you get worried on the safety of such a high Obinutuzumab dose protocol? Can you please comment on this thing of being immunosuppressed within the protocol with a potent B-cell depletion that was done during COVID?
Yes, this was such a great question, Raquel. And, I think, of course, we always have to be very careful to analyze the safety of any new agent while we also think about the efficacy, right? They go hand in hand, the efficacy and the safety. And I think what was interesting about this this trial, and I would love to hear Rich's comments on this as well. Going back to the phase 2 Nobility, we didn't see a signal in terms of more serious adverse events with Obinutuzumab versus placebo. In this trial, we did, right? So this trial, there were more serious AEs and infectious AEs with the Obinutuzumab group compared to the placebo group. And there were more COVID-19 related adverse events as well. And so I think we have to, of course, look at that. I think what was quite striking in this trial was that it was able to be conducted and completed despite the fact that the pandemic was occurring. I think, if I remember correctly, this trial began to enroll participants 4 months into the pandemic, which is incredible that this trial was able to continue and enroll. So we have to think about that. We have to think about the fact that this trial was enrolling participants before we had vaccines approval for COVID-19, as well as the effective therapy with monoclonal antibodies. So all of that has to come into play. But yes, I think we have to always consider safety. I think what was interesting in this study as well, one thing that we worry about with anti-CD20s therapies, including rituximab, over the course of time, we can see hypogammaglobulinemia.
Yes.
And in this trial, there were some results that are shown in the supplementary materials of IgG level, and there was a decline in IgG initially, but then we started to see the IgG kind of recover over time, which is very interesting, despite the fact that there were recurrent doses of Obinutuzumab given. So I found that to be very interesting. And I think we can look more deeply into that, I'm hoping, as more data are released and analyzed from this particular trial. But we have to look at that. And of course, think about safety, and then questions about, are there differences in safety? We don't have the answers here, but when we think about using these agents, this agent in this trial was used in combination with background mycophenolate at a lower dose, right? So the mycophenolate was 2 to 2.5 grams a day. The glucocorticoids were also used in a lower dose than traditionally have been used in lupus nephritis trials. So I think those are all moving in the right direction. I think as we begin to reduce background therapies, the hope is that we will be able to reduce infectious complications. But I think all of these things have to be taken into consideration. And again, emphasizing the importance of vaccination before one embarks on really any immunosuppressive, but particularly anti-CD20s, which clearly impact the ability of us to mount immunologic responses to vaccine, at least the humoral responses to vaccines.
Thank you. Richard, what is your comment on the safety part of the results?
Yeah, so studying an immunosuppressive during COVID is far from optimal. We ran into this problem with the anifrolumab long-term extension also. And so in this study, and it's in the supplementary material, there were fewer COVID-related events in the second half of the study, which just speaks to improved resources, better knowledge regarding how to deal with COVID. The experience with rituximab and COVID is that humoral responses are compromised with B-cell depletion, although cellular responses seem to have been preserved. But if you throw in mycophenolate and throw in some steroids to the equation, both arms of the immune system are quite affected. So certainly vaccinations are key, and we're not just talking about COVID vaccines. I am a big believer that all of my lupus patients get the flu shot. They get immunized against pneumococcus, shingles with Shingrix. RSV, I'm not sure just yet, but why not?
Going back a little bit because we are always comparing rituximab and obinutuzumab. And on the beginning, you already spoke a little bit about how this drug was made. And do you think, Richard, that the greater clinical results in this trial is because obinutuzumab is different from rituximab, or do you think there's other variables that are here taken in account?
No, well, my own personality is I'm not a more is better kind of person.
Okay.
I think in this situation, more is better, and we're talking about more B-cell depletion. So how is that achieved? And let me just tell you a little bit about that. So unlike rituximab, which is a type 1 anti-CD20 monoclonal antibody, obinutuzumab is considered a type 2 antibody. And it has a different binding configuration, which enhances antibody-dependent cytotoxicity. It enhances antibody-dependent cellular phagocytosis, and it causes direct cell death more so than rituximab did. But I'd say the major change was that it was defucosylated. I mentioned this before, which also enhances ADCC, and it enhanced it by about 100-fold. So it's known before even starting the Nobility trial that this engineering led to greater B-cell depletion, both in vitro as well as in B-cell malignancies. In fact, there was a small study, though this was done a little bit later, but it proves the point. There was a small study in a couple dozen patients undergoing kidney transplant who had previously received obinutuzumab because of prior sensitizing events. They harvested lymph nodes at the time of transplant and evaluated the B-cell populations. And basically what they found was very significant B-cell depletion in the tissues, which was not seen in a very similar experiment with rituximab. So I would say, yes, obinutuzumab's success supports the more is better in reference to the degree of B-cell depletion and clinical response.
So with this that you told us about and the difference in lymphomas, do you think that when the time comes that obinutuzumab is going to be approved to lupus nephritis, we should no longer use rituximab?
Yeah, that's been the case in my division because this to obinutuzumab. And so for anybody thinking about rituximab, at least in lupus nephritis, then we go with obinutuzumab. And we've treated close to 60 patients off-label, half of whom had lupus nephritis, others had myositis or ankylosing who may have been allergic to rituximab.
Okay. And I think that we should move now to some of the tricky parts of the trial. That is the subgroup analysis that is really challenging to interpret. How can you explain, Richard, that men had a higher complete renal response in the placebo group?
Frankly, I can't. The interpretation of subgroup analyses has to be approached with caution since they're always underpowered. They can be hypothesis generating, though I don't have a hypothesis for this situation. The most unexplained group, was the difference between females and males. So where the former, the females, had complete renal response rates of 20% in the placebo group, 47% in the obinutuzumab group. That's a nice effect size. But if you look at the males, it's the polar opposite. The males had complete renal response rates of 67% in the placebo group and 43% in the obinutuzumab group. So it was a surprising result. It's unexplained, but I think it's just a quirk of the subgroup analysis, basically dicing the cohort into slivers. And certainly the proportion of men was significantly lower than females.
Yes, Maria, do you have any additional thoughts on this?
I agree. When I thought that too, when I saw these data, I saw the outlier of the males, and I agree. I think that most likely this is just explained by the fact that it's a spurious result, that the sample size is tiny, right? 21 males in obinutuzumab and placebo. So 15% of the total trial population, around 15%, were men. So you start to see these kinds of unusual results sometimes when you start to have these small sample sizes. I can't think of a biologic effect that would have led to this difference. Again, with just this higher placebo response rate in the males, I think it's likely due to just a small sample size.
Yes, and it should not be interpreted to use clinically, and hopefully the regulatory policies won't have this in account for anything. Another subgroup was the ethical and geographic differences. Richard, I'm absolutely sure you all have scrutinized completely every single result. Do you think that the differences can be clinical meaningful?
No, I just think this gets back to the comments I just made about subgroup analyses and whether one examines race or the geographic region. The point estimates in each case favored obinutuzumab. But I don't see regulators acting on these subgroup analyses. I'd be very surprised.
Okay, thank you, Maria. One of the things is that I don't know if it happens in USA, but actually after FDA approves, then EMA also see the results and approves, and generally each country has another regulatory policy, and at least in some of European countries, the regulatory entities go and seek in the trials what are the subgroups that most benefit from the drug, and they tend to first approve to reimburse those groups and not all the population. That's why I was asking you that, Richard. And Maria, one of the subgroups that really benefit the most were the more active groups, like the difference between class 3 and 4 and having or not having class 4. And what are your comments on this difference, on the severity of lupus nephritis and classes of lupus nephritis?
So I agree with what Rich said, that I think what was reassuring when we looked at these pre-specified subgroup analyses was that we saw numerical efficacy of obinutuzumab in each of them, numerical efficacy, except for the male subgroup that we just talked about. And I think when we look at this globally, that is reassuring. I think that, again, we look at these subgroup analyses as being hypothesis generating. They give us additional information, but we can't put our weight behind any specific one of these because, again, the trial was not powered, right, to assess efficacy in these various subgroups. And so we have to look at them with a grain of salt. And they can give us, again, information, they're exploratory. So in that spirit, when I look at these, and as you said, I think what was very, again, reassuring to see was that in those participants that would be considered to have higher markers of disease activity, right? So the elevated anti-DNA, low complement, the class 3, 4, even the plus or minus 5. I thought that was very interesting. So in the subgroup in which there was concomitant class 5, clear numerical efficacy. And that's interesting because often the patients, in my experience with the concomitant class 5, can be more challenging to treat, take longer to see an effect, right? Because of the class 5 lesion. So that was very interesting and helpful. And then I think another important subgroup were those with the high levels of proteinuria at baseline, the greater than equal to 3 grams at baseline, and clear efficacy there. And remember that in the Bliss lupus nephritis trial, which was the belimumab phase 3 trial, in that subgroup with the high level of proteinuria at baseline, the numerical efficacy was not seen in that subgroup. And that was a subsequent publication to that trial, a post hoc analysis. And so I thought that was very interesting and, again, reassuring across the board that this mechanism of action will be useful, I think, in these various circumstances, in these various subgroups. So I think it just gives us reassurance in terms of how this agent can be used.
Richard, I know you've seen the same and explored it. And there's another thing that I don't know what was your intention by doing this subgroup analysis, but the difference between those who had a previous lupus nephritis and lupus nephritis for the first time. What are your comments, Richard, on this?
Yeah, so it's true from the subgroup analysis that the response rate to obinutuzumab was higher in the newly diagnosed subgroup than in the previously diagnosed subgroup. It was 52% versus 43%. So that intuitively makes sense to me. But an important point is that those with a prior history of lupus nephritis were not necessarily refractory to prior treatment. They were just reflaring. So at this point, that's all we can say is that they were once again flaring. But if we look at the placebo response rates in these two groups, the complete renal response in the previously diagnosed group was some 11 percentage points higher than in the newly diagnosed subgroup. And this is contrary to what I would have expected, and it led to a considerably smaller effect size in the previously diagnosed subgroup. So maybe just a quirk of the relatively small sizes of the subgroups. Anyway, the bottom line is I would not distinguish between the two groups, certainly, when it comes to using this drug in practice.
Yes, actually, one of the recommendations is to use B-cell depletion in refractory lupus nephritis. So it tends to be thinking about it, it should be suspected that it would work not only on the first lupus nephritis, but also on the others. There's another big clinical important outcome. It's the eGFR slope data. And I know that we shouldn't compare trials, but can you compare this data between Nobility and Regency, and is there a way of extrapolating this outcome from Bliss LN trial and Aurora? Richard, have you think about it?
Yeah, so I agree that eGFR slope is gaining traction as an important metric, certainly in non-lupus nephritic diseases. But The difference between the two obinutuzumab studies, Nobility and Regency, was not that great. In fact, I calculated the delta was about 1/4 of a point when comparing the differences in eGFR slopes between drug and placebo. But more importantly, in Nobility, the calculation was performed starting at week 12 and ending at week 104. Whereas in Regency, the calculation was performed between baseline and week 76.
Okay.
Whether the variation in the evaluated intervals makes a difference, we'll know in time when we have more data after week 76. And then if you start talking about comparing to other studies like belimumab and voclosporin, yeah, we shouldn't be doing that, but I did it at least for comparing to Bliss-LN. And the deltas between treatment and placebo were not that different in the 3 studies. I didn't look at Aurora. Aurora is even more difficult because of the mechanism of action of voclosporin and the fact that over time, a lot of the patients on voclosporin reduced their dose, so their eGFR went up sort of, I guess you could say falsely. But again, back to Nobility, Regency, and Bliss-LN, the deltas were a little over 4 in Nobility and a little under 4 in Regency and about 3.6 in Bliss. So they're all somewhat the same, even with the differences in the study design, the differences in the intervals that were evaluated.
It's good. It's when there is not as much as a difference with placebo, it looks like the standard of care also prevents the slope of the GFR, even though we can't really compare between trials. Maria, what do you think is the most important secondary outcome of this study?
Yeah, that's an interesting question. I think for me, so I think we just spoke about the change in eGFR, which is a very important outcome because after all, what we're most interested in is preserving long-term kidney health, right? Preserving kidney function over time. So the eGFR endpoint over time is critical. And we saw, as we just discussed, that there was, a signal there with obinutuzumab over placebo. I think if I had to choose, I would choose the complete renal response at week 76 tied to the prednisone taper. I think to me that is a critical finding. And just to remind everybody, the first key secondary endpoint was CRR at week 76 with prednisone taper to less than equal 7.5 milligrams a day. Between weeks 64 and 76, 42.7% of the obinutuzumab group met that endpoint versus 30.9% of placebo, with a statistically significant difference there. And that to me is very helpful because, again, we're moving into this era. We've moved into this era now where we're using lower doses of glucocorticoids. We all know about the importance of using glucocorticoids judiciously. We still have to use them in lupus nephritis, but we're using them at lower doses. We're starting with pulses and then a lower starting oral dose with an expeditious taper. And the taper in this particular trial was to try to get down to less than equal 7.5 milligrams a day by week 12, and then 5 milligrams a day by week 24. And by the way, we have those same recommendations in the updated ACR-LN treatment guidelines, the same sort of regimen. And I think we always want to be able to see efficacy of a new therapy, in this case, obinutuzumab, at the same time that we're able to taper down glucocorticoids. And that was shown here. So I think that's a very important finding, again, that we're seeing efficacy of this new agent, despite the fact that we're tapering this steroids, which we know are toxic over the course of time.
Thank you. Richard, any words on the intercurrent events data?
Yeah, I must say, when you read the article, you need a glossary. So you need to keep straight what is meant by intercurrent events, what is meant by renal-related events, what is meant by treatment failure. But the bottom line is intercurrent events are essentially pre-specified, pretty much kidney-specific adverse events. And
Okay.
of the intercurrent events, which were more than twice as common in the placebo group, the noteworthy ones were clinically significant sustained worsening in UPCR or eGFR onwards from week 24. And just to get some idea of the numbers, meeting those metrics, in the obinutuzumab group, it was about 3.7%. And in the placebo group, it was a little over 16%. So that's a very significant difference.
Yes.
The other part of intercurrent events that drove this was the need for rescue therapy. And for the obinutuzumab group, about 6% needed rescue, whereas for placebo, it was actually almost 3 times as high, almost 18%. So the intercurrent events was, I think, the 4th secondary endpoint. And the rule is, the statistical rule, which I never quite understood, is that as soon as you hit one secondary event that's not statistically significant, you really can't or shouldn't evaluate with rigor the statistics on the ones below that. But the top 2 out of the 6 were statistically significant. The third one, which was, I guess, eGFR, came close, but did not hit statistical significance. But yeah, there was a very significant difference in the event rates that I just mentioned.
Yes. And you just explained why it does not appear on one of the main tables of the results on the paper, because I was that not statistically significant? So they didn't present the results? What's going on here? Thank you. Yes, I don't understand why, but okay, the rules are there. Thank you for that.
Well, in fact, it was at an ACR meeting. I was on a panel and I mentioned something about the rules. It was in relationship to the anifrolumab problem that we had. So recall, TULIP 1 not successful, TULIP 2 successful, and the phase 2 anifrolumab was quite successful. And then you have all the secondaries. So the totality of the data spoke to a successful outcome for anifrolumab. But if you're held to the primary, of the pivotal not being successful, and you rely on the secondaries, why have a hierarchical order? Why not just kind of put them all together and say, oh, you have to achieve, like 60 or 70% success on your, 9 primaries and secondaries. But anyway, a discussion for another day.
So, talking about rules, what else is needed for an FDA approval of obinutuzumab?
Well, actually, I'm not sure anything. The 2 trials, Nobility and Regency, will give the FDA a lot to read. And I'm not aware that additional data or additional trials will be needed. I mean, there is another trial that's being done, and that's obinutuzumab in SLE. But that will not bear on an approval for lupus nephritis.
Okay. Maria, let's assume obinutuzumab is going to be approved. Where would it fit in your lupus nephritis treatment?
Yeah, I think this is the $1 million question, right? To think about how now, looking ahead, if this is approved, how do we fit obinutuzumab into our therapeutic paradigm for lupus nephritis? And just taking a step back and again thinking about where we are now with the 2024 American College of Rheumatology Lupus Nephritis guideline for the screening, treatment, and management of lupus nephritis, in which the proposal now is to use combination therapy, which we're calling triple immunosuppressive therapy, which means glucocorticoids plus 2 additional immunomodulatory immunosuppressive agents. And right now, as we all know, we have voclosporin approved and we have belimumab approved, and those can be utilized within this triple immunosuppressive therapy. So now we have to think about, let's say obinutuzumab is approved. How do we fit obinutuzumab in? And I think that it would be added in as another option for what we call triple immunosuppressive therapy. Obinutuzumab in this REGENCY trial was used in combination with background mycophenolate mofetil, again at lower doses, as well as glucocorticoids. So it's going to fit into that rubric of a triple immunosuppressive therapy. And I think the challenge that we have in lupus is we don't have either the blood biomarker or the tissue biomarker yet that will readily tell us which patient will respond to which mechanism of action. Which of these triple therapies will be most effective and safest in a given patient. And that's what we have to get to, right, is trying to understand what's happening molecularly in the tissue or hopefully in the blood, because it's easier to sample the blood. We know that coming down the line, we have urinary biomarkers, which I think are going to be helpful in helping us to understand how active lupus nephritis is. However, those markers still are not going to specifically tell us who is going to be most responsive to a B-cell directed therapy versus another mechanism. And that's what we have to get at. So I think this is the challenge. And I think that, however, how do we actually make rational choices in this time when we don't have all the data, we don't have the biomarkers? We still have to make decisions, right? Because we are taking care of our patients in clinic and they're coming to us, and we have to figure out a way to make informed decisions and engage in shared decision-making with our patients. So I think we're going to pull from data such as, I think, and when I look at the data from this particular trial, I'd love to hear your and Rich's opinions too. But I'm reassured again by just going back to some of the data that we saw, that there was efficacy in these high disease subgroups, patients coming in with high levels of proteinuria, with the high DNA, with the low complement, with the concomitant class 5. I think all of these things are helpful, and we see some differences there with our other approved agents of belimumab. I pointed that out earlier. I think what's interesting, one thing I want to see, I was going to ask Rich about this that I did not see in this paper, and I would like to see it in subsequent analyses, is time to reduction of proteinuria, right? Because we know that the calcineurin inhibitors, one of the benefits of those, that class of agents, just based upon their mechanism of action with direct effects on the podocyte, is we see a rapid reduction in proteinuria, which we know is beneficial to overall long-term kidney health. Are we entering a situation where the calcineurin inhibitors are helpful early on to get the proteinuria down, but then we have to use B-cell directed therapies for more long-term efficacy? But again, I would love to see the kinetics of proteinuria response with obinutuzumab, right? That's going to be interesting, and I would expect that would come out in further analyses, and Rich might even have the answer for us right now. But I think that to me is going to be intriguing to think about how to position these agents. And the last thing I'll say, when I think towards the future, is thinking about, how can we start to use these targeted therapies in combination? Because right now we talk about triple immunosuppressive therapy, but it's still kind of the conventional therapy, the MMF, right, in combination with our targeted therapy. What about thinking down the road about combining targeted therapies, sequencing those, and trying to peel back some of the non-specific conventional agents? That's the other thing in the back of my mind.
Richard, I'm going to ask you exactly the same question, and I'm going to add a question for both of you. Are we going to get rid of cyclophosphamide in the future? And taking account that belimumab, voclosporin, obinutuzumab, Richard, how do you think through all this, and taking account also those who have only lupus nephritis versus extra-renal activity also? And how are you going to use obinutuzumab in the long term? Are we going to do 6 infusions and every 6 months after that? what is up on your clinical mind, Richard?
I was hoping we'd run out of time and I could avoid answering that question. Yeah, I mean, there's no question we had to figure out how to use these drugs. But if you go back not that long ago, our toolbox was empty. And now we have choices, but with choice comes uncertainty and confusion. But each of the drugs has its own merits. Belimumab has been around the longest, a great safety profile, and it's a great drug for maintenance. Voclosporin reduces proteinuria quite rapidly, as Maria said. And we're appreciating more and more the value of proteinuria reduction. And obinutuzumab, based on the subgroup analyses, as you pointed out, seems to benefit those with features that represent the most active patients. And, I, like Maria, can complicate this issue even more. So what about sequential use of these drugs? Or what about monotherapy? Could obinutuzumab, with maybe a little steroid up front, be used as monotherapy? So we have to figure that out in the future. Now you asked some other questions about, would extrarenal manifestations sway me? What about long-term dosing? All I can say is obinutuzumab is being studied in an SLE trial, so we'll see how extrarenal manifestations fare. And as far as long-term dosing, I'm not sure how clinicians will use obinutuzumab long-term. I mean, we will get some data from the extension study, but the situation's almost reminiscent of where we were with rituximab and rheumatoid arthritis. Do you keep giving it every 6 months, or do you wait for the patients to flare? But I think clinicians will probably feel uncomfortable keeping patients B-cell depleted long-term. But I do have some patients who were in the Nobility trial and who received obinutuzumab during that trial. And it's now, oh my gosh, it's like 7 years later, and they remain off all immunosuppressives, and their nephritis is in remission. So that's another thing that needs to be looked at, because when our clinical trials end, the clinical trials end, and there's really no long-term follow-up.
Yes. And this brings me to the final provocatory questions for both of you. CAR T-cell treatment is really rising attention of everyone, making us hope of a cure. And actually, it's almost based on a really, really profound B-cell depletion. So do you think that obinutuzumab, as you said, Richard, that you have a patient that is free of drugs and in remission, do you think this strategy can be a safer CAR T cell depletion treatment? What are your comments on it?
Well, I do think the B-cell depletion reported so far with cell therapy is deeper than with monoclonal antibodies. But the real question is whether total elimination of B cells is necessary in lupus, like it is in lymphoma. And a lot of these questions remain unanswered. We don't even know what the best target is. Should it be CD20? Might you do better with CD19 target, or go a little bit more distally and target BCMA or CD38? We don't know. And what is the best method? Is it cell therapy? Is it a T-cell engager? Is it a monoclonal antibody? So we had to figure all this out, but it's not easy to figure out. There are so many companies wanting to try to figure this out, but it's hard to enroll patients into all of these studies. But regardless of these challenges, there's no question the future is bright for our patients with lupus. We've come a long way, and there's no doubt that the outcomes will improve over time.
Maria, any final comments?
I agree with everything Rich just said. I think this is an exciting time for our patients living with lupus nephritis, that we have these options. Not only are 2 approved therapies, but now we have obinutuzumab, and then we have this plethora of other agents, and even just thinking about agents targeting B cells, as Rich just described, We have agents that target B cells at different stages of development, right? Depending upon which antigen they target. Is it CD19? Is it CD20? Is it anti-CD38? Is it BCMA? So I think that the question again for the future is going to be, how do we know which B cell subset is most pathogenic in the patient that's coming to us? And that's where I think the field is going to be headed. But I think we are going to have options. And I think specific to the question of cell therapy, I think, this is a very exciting frontier, but we're still very early, aren't we? We're awaiting more data from the clinical trials that are being conducted now, the larger phase trials. There's still phase 1 and phase 2, but still more data needs to come with a more diverse patient population than some of the initial reports. So we're waiting for those data. I think just inherently with the autologous CAR T cells, just to take an example, it's such a cumbersome process that I think if looking down the road, if one had an efficacious monoclonal antibody such as Obinutuzumab and other monoclonals coming down, or the bispecifics as Rich mentioned, that are going to be easier to administer to our patients, I think that's going to be an advantage, right? But there are going to be those patients who, and we all see them in our practice, who are unfortunately incredibly refractory to many of the therapies that we try. And I think having options such as cell therapy will be beneficial, although we still have to see what the ultimate benefits are just from the monoclonals. I think putting them,
Yes.
I think it's going to be very interesting to see. The last thing I'll say is just mentioning the question or the issue of kind of this immune reset that people talk about, right? That people talk about with cell therapy, and just building off what you and Rich stated, back when I've used rituximab for off-label for refractory lupus nephritis patients, as was described by Rich, I have seen these long-term, even with rituximab, 6-7 years off immunosuppressive therapy, still B-cell depleted, which is a little bit scary. Why are they still B-cell depleted after all of that time? With the cell therapies, B-cells repopulate in about median of 100 days, right? So has there been an immune reset even with the old anti-CD20 or rituximab? So we still have so much to learn about how to position these therapies, but I think the future is bright, and it's wonderful to have these options now to be able to offer our patients.
Thank you so much. It's the first time we're doing a podcast only discussing one trial, and I do think it was really exciting discussion. So Richard and Maria, thank you so much for joining me today and sharing your insights of the REGENCY trial. And to our listeners, we hope these discussions have helped you gain more of an understanding of lupus and even lupus trials and the developments into 2025. Thank you so much for listening. We hope you enjoyed this podcast. Please leave us a review, which will really help us in reaching as many of your colleagues as possible around the world. Join us again next month for more discussion, key issues in the management of lupus.